Hi everyone, so my name is Takeshi Nakano. We are working on the plant biology. Especially we are interested in the plant growth mechanism. To reveal the mechanism, we are using the plant chemical biology. Let's go to see our laboratories. This is the general mechanism of photosynthesis. Plant is a photosynthetic organism. From CO2 and sunshine, a plant produces oxygen and carbohydrate. The oxygen supports respiration of animals, and fixed carbohydrates support food chain on the earth. By this function, we can say plants are supporting almost organisms on the earth. In any cases, the animal uses peptide type hormone to regulate animal functions. As plants produce carbohydrate by themselves, plants have created and modified carbohydrate type chemical to regulate plants themselves. The evolutionarily produced plant chemicals are widely used by plants, and humans use this chemical for the medical science. Then, in recent years, number of natural plant chemicals are predicted to be over 1 million species. So many species. Based on this idea, we are trying to use chemicals as a useful tools in plant biological research. Right side is a genome and protein. It's a currently famous molecular biology. Left side is a natural and synthetic chemicals that have a bioactivity onto plant growth. New strategies to use chemicals for revealing unknown mystery of biology, and now called as chemical biology. We are trying to reveal the plant growth mechanism by the plant chemical biology approach. For example, by our plant chemical biology, we have identified several kinds of new factors that are working in plant hormone brachiosteric signaling. Okay, let's go to see our laboratories. This is Arabidopsis. This plant is most famous in molecular biological field all around the world. By combinational research of the chemicals and the Arabidopsis, we are trying to identify the mutant that shows the different development in the plant growth. This is the wild type. This is a mutant for the plant development. If we identify the mutant that shows plant growth defective phenotype, we try to isolate genes that cause mutant phenotype. Next, when we could identify the new genes, we are analyzing the molecular function of the genes by molecular biology, biochemistry, and chemical biology approach in this room. This is a confocal fluorescence microscope to analyze the protein dynamics of each novel factors in plant cell. We are attaching GFP named fluorescence protein to our novel factors. The fluorescence image can suggest where our factors localize in the cell and are moved by chemical stimulation and so on. Finally, if we can find very good gene, it can high effect to the plant growth, we are transformed to the rice. If we can find this GMO rice can show the plant growth promoting activity or stress resistance activity. This gene should be contribute to the future agriculture or repairment of the environmental stress condition of the earth. This is our research goals. If you are interested about our research, please contact us.